Together with Alexandru, we will present now diffusion for molecule generation. So now what is going to happen? First, I will have an introduction with why and how molecule generation is done. And then I will go through chirality and isometry groups that are important concepts. And then I will have a brief diffusion basics. And then diffusion on point clouds. And then last of my part will be problems with E3 invariance. And then Alexandru will take the latter part and talk about how to actually use fields and new representation for molecules, and then talk about the challenges and solutions, how to handle these, and then empirically confirm these results that the theory I will present here first. Okay, so why molecule generation? So we want to make some molecules for medicine, and historically those have been found from nature, but now we can use generative models to generate an initial set that we can then in silico filter and then further filter in the wet lab. And then we can furthermore do conditional modeling. So taking some condition like a binding pocket and have a better initial set. And using this kind of automated pipeline could give a lot of savings because drug discovery is extremely expensive. But first, the models need to actually generate some sensible molecules. So they need to adhere to basic chemistry rules like the octet rule, and then they need to be stereochemically feasible. So the 3D conformation has to be something that can actually exist. And the models need to scale to larger compounds, both computationally, but also when larger compounds, they are more complex. So the models need to also know how to model these more complex molecules. So for this, as Martin told, that these molecules have to be represented in some good ways, so that the model can actually use important features. So a little bit like just Martin said, that the smiles, which is this string representation, it kind of doesn't make sense because there is no natural start and end or ordering of molecules. And also in the smiles representation, one molecule can have many very different smiles. So the model would have a hard time adding both of those representations to the same molecule. Then also graphs are kind of good, but these do not consider the 3D conformation. So there is also a deficiency there. And then we have point clouds. So these are quite good. So these are just points living in the 3D space. So these can handle the 3D conformations. And then the bonds can be either modeled directly or then inferred from the distances of the points that can be atoms. Then lastly, we will have fields. And this is amazing. And Alexandru will present more of the fields approach. So then you can actually, for the generation of these molecules, you can, given a graph, generate the atoms for the graph and get the molecule, or you can generate fractions instead of atoms. Or then you can just create the connectivity graph, connect different atoms. But the thing we will consider most here is to generate molecular graph and the 3D structure, so the full molecule. And now one important thing is chirality. So this is handedness, and this is a spatial property that the mirror image of a molecule cannot be superposed on the original one. So it's exactly like your right and left hand. You cannot rotate your right hand or translate and rotate your right hand to get your left hand. you actually have to have a reflection. And chirality is quite important in biology. For example, like metabolization is dependent on the chiral enantiomer, and also here the ibuprofen is only effective in one of the enantiomers. So chirality is an important feature. And kind of related are the mathematical isometry groups, where we have the Euclidean group, which is rotations, translations, and reflections. So if some function is invariant to E3, then it cannot distinguish between the two enantiomers. So kind of a better way is to have SE pre-invariant methods, so that rotations and translations do not make a difference, but then reflections do. So this would be like really nice for all these molecular generation methods. I will come back to this later. So first I will show some diffusion basics. So diffusion is, well, quite a good generative approach, used quite a lot. So we want to model some data distribution of interest, and more specifically, we want to sample more from it. So how do we do this? We take a noise distribution, here a Gaussian, and then we want to couple these distributions. So going from the distribution of interest or the data to the easy distribution is quite easy. We can just gradually add Gaussian noise. This is called the forward process. So here we can choose that do we add a lot of noise in the beginning or middle or end, and then to what sort of Gaussian we come. So this is kind of the noising schedule that we can choose. And then eventually we want to learn the reverse process. So to take noise and then make this back into the distribution of interest. And yeah, for example, images, this is quite a basic application. And we are using the DDPM formulation. And here we have x0, which is the clean data point, and x1 is the total noise data point. And here Q denotes the forward process. So the noising process, this is defined by our noising schedule. So we know this is nice Gaussian, and the P is the reverse, and this is something we want to learn. So at any time point now, between the 0 and 1, we would like to have the same distribution if we go from the forward and go for the backward. So one could simply take now the forward here and the backward, and then match them, but this would be kind of computationally unfeasible, because you would need to have two random variables and make a Monte Carlo estimate of that. So instead, you can match it to the posterior of the forward, and using a clean data point, and this will then also be tractable, the posterior, and now we can actually match these, which in a computationally feasible way. And the reverse process can then be parametrized by taking the posterior, and instead of an actual clean image, we take a neural network and predict a clean image based on the noisy image. And now we can have a variational lower bound and make a loss. And now the main part of this loss would be the matching of the posterior and the reverse. And these would be then Gaussians with the same standard deviation. So we can further simplify this loss to just a squared error. And then also in practice, it is easier to learn just the noise, not the clean data point. So this is kind of mathematically equivalent, but it's in practice a lot better. Yeah, I think I'll skip this, but there is a neat way of doing conditioning for diffusion. And that was classifier-free guidance. But yeah, okay, so diffusion on point clouds. So there is this EDM, which is like a seminal work for this E3 equivariant molecular generations. So these are then using point clouds, so points in 3D space. And this is then parameterized by a graph neural network that takes distances as input. And then yes, these 3D coordinates are then diffused and modeled, and based on the clean positions of the atoms, then you can determine the bonds. So here it's important to note that both the positions and thus also the bonds and also the types of the atoms can be changed basically at every time. So you can never know that this is for sure a carbon. Yeah, and here is an illustration of the denoising process. Okay. Now this is actually like kind of nice. These point clouds having the E3 invariance because of the rotations and translations, but there is this problem that if we have a E3 invariant distribution, then the enantiomers will appear at the same probability. So this is quite bad. For example, if we want to model a data set of highly metabolizable molecules, and this is very dependent on the chiral configuration, this is actually not good if we have a E3 invariant method, because then it will just randomly 50-50 choose which enantiomer will come out. And actually, so this GNN in EDM and many other point clouds is E3 invariant. So this also makes the conditional equivariant, and furthermore, this will make, given that the prior is invariant, this will also make the probability distribution that you generate invariant. And this means that, yeah, it cannot consider the chirality, the current point cloud methods. Okay, so now can we fix this and make it SE3 invariant? So now we can take a function that is SE3 invariant, and if we actually have a function that takes an object of m n-dimensional vectors, but m is less or equal to n, then actually the function output will be E3 invariant. And yeah, just to remind that, yeah, we can do any rotation and the function will be the same. But now actually, because the dimensionality, any reflection can also be formulated as this rotation. And we can look at this example in 3D space, where we have these 3 points. And we can reflect them, but we're reflecting on this yellow plane, but the reflection is actually exactly the same thing as the rotation around now the line L here. So now if the function is SE3 invariant, it will also be E3 invariant for these 3 points. So this means that if we have just 3 points, you cannot get an SE3 invariant feature. Okay, but we still want this. So if we have now E3 invariant features and a function that eats those features, then it cannot be anything else than E3 invariant. But okay, so how can we get something that is SE3 invariant but not E3 invariant? So we have one suggestion, which is the determinant. So we have 4 points, and we subtract one of the points from rest of the 3 ones, and then we take the determinant. So the determinant will have the value and the sign of the volume and the sign of the volume of this tetrahedron, and the sign will actually tell the chiral enantiomer. And then actually, if we have 2 of these function outputs, if we do not have the same 4 points, they will actually be independent of each other. And then now we say that, okay, if we want to have one of those point clouds that for any time point, we actually cannot know what the atom type is and also the exact connectivity, then we need to have O to the n to the power of 4 of these features. And this is kind of bad, meaning that it's kind of hard to actually create this point cloud method that would actually be SE3 but not E3 invariant. And that's what I wanted to say. So now Alexandru will present a solution.
Hello. Yep, I am Alexandru Dumitrescu, and I will continue by presenting one of our recent works published this year at ICLR, entitled E3 equivariant models cannot learn chirality, field-based molecular generation. So I will briefly recap what Dani said. We want to present generative drug-like molecules, generative models for drug-like molecules, because this can be a means of more efficiently exploring the vast space of molecules, and this might help us in the near future to accelerate drug discovery. And then we know that the efficiency of drugs is highly dependent on their three-dimensional conformations, and therefore much of the prior work has focused on these point clouds that model directly the atom coordinates and atom types, and that they are predominantly E3 invariant, so invariant to any global translations, rotations, or reflections of the molecules. And there is this problem that chiral molecules are not reflection invariant, and that reflections of molecules that contain chiral centers will create enantiomer pairs which have distinguishing properties, and that E3 invariant methods cannot capture or distinguish. And so furthermore, we also argue how chiral adaptations are quite challenging to develop while keeping rotational invariance. And then here is the same image with ibuprofen that is only efficient in its S form. So we propose diffusion models on fields, and concretely, we define scalar fields in the three-dimensional space for each atom and covalent bond type, which have the following form. So they are sort of unnormalized Gaussian balls with specific field parameters, gamma and standard deviations. And they, of course, depend on the atom positions M. And kind of a depiction of these continuous fields is on the right-hand side, where we present this benzene, which is a planar molecule, and therefore we can only show a z=0 section of the field. So the field is 3D, but now we only look at the section of it. We form our inputs by evaluating these fields at specific locations G, and we do this for both the atoms and bond fields. And we select equally spaced locations G, therefore creating fields like this image. And then, yes, so the final input representation is the concatenated evaluated fields for every atom channel and every covalent bond channel.
Alexandru, how are atoms and bond fields coupled? Obviously, one is heavily dependent one on another.
Right, so I'm not exactly sure what you mean by coupled, but I guess they are separate fields, but they, of course, influence the neural network activation. So the neural network, I will describe later how the neural network works, but the neural network sees both fields and atoms at the same time and should hopefully learn to create reasonable molecules. And as we will see later, to a good degree they are.
So the neural network, in other words, learns you cannot have a bond if you don't have two atoms on the right positions.
Yeah, so it is considerably better than at least that. So it's never generating bonds at where there aren't atoms. Yep. Okay, so now we will go through the architecture and the invariances of our architecture. So since we have equally spaced grid locations G, we can use the three-dimensional adaptation of a unit. And just briefly describing what this does. So we have multiple downscale and upscale layers for various resolution levels. And then we have sort of similar to skip connection layers that concatenate the downscaled hidden representations to the upscaled hidden representations. And the intuition on this is that the architecture can capture both the global features of the molecule as well as the local features for the low and then high resolution levels, respectively. In terms of the E3 invariances, we have good translational invariance, inductive biases, because this architecture has most of its parameters in convolutional neural networks. Then we have no reflection invariance, which is, of course, a good thing, as we can distinguish enantiomers, but we do not have rotational invariance. So what we propose to mitigate this issue is that we define reference rotations as rotations about their principal components. And again, the same benzene molecule is depicted here. Now, going through the diffusion noising process. So we use a variance-preserving noising process defined by some alpha parameter that gradually removes the signal U0, which is the clean input field. And then the noise added at each time step is directly defined by the alpha t schedule, which defines how fast the signal is removed. For alpha t, we use a generalization of the cosine noising schedule with this new parameter, and setting nu equal to 1 and nu equal to 1.5 will result in generating bonds faster or equivalently destroying atoms faster. And this is an example of a generated molecule. So here we show 200 highest values across all the bonds and atom type channels respectively. And you can see, probably also related to the question earlier, that they are sort of, even though we don't restrict the model to generate specifically bonds at locations where there are atoms or vice versa, then of course these are still reasonably well coupled in a sense. Then after we have generated the fields themselves, of course, we just have some values in the three-dimensional space. We would like to actually retrieve the molecular graphs from those fields. And first, to do this, we look at the atom fields. And we detect field locations that have high enough peaks and assign the number of atoms and rough locations M for each atom type based on a threshold-based algorithm, which is a very efficient, relatively simple algorithm. After this, our rough initial atom positions, of course, are not good enough. We want to actually optimize them. So what we do is essentially we fit the functional form of the atom fields to the generated values. So the bolded UAX here is the, or represent the generated field values at locations X in G. And we would want to minimize the squared error loss to the functional form of UAM of X for all the locations, for all the atom locations M. So we optimize the atom locations here. Then to initially extract bonds, this is quite a bit easier. We simply can look at only the mid positions between any two atoms, so Mi plus Mj by 2, and determine whether or not a high enough value is within that region. And for each Mi plus Mj by 2 value which is high enough, we assign some component gamma ij. And then this time we optimize for the gamma values. So you can see a very similar optimization function here. So the arc mean over the gamma this time, which also minimizes the squared error loss between the functional form of the bond fields and the generated bolded UBX. And on the right-hand side image, we have an example of a QM9 molecule with noised out fields. So the procedure is generally robust enough that if you don't actually add noise to the fields of a data molecule, the extraction will just be correct. So here we also added on top of the original field some noise. And then we can see that the bond marked with X should not be there. It should not be there because there are no actual Gaussian peaks at that specific location. And correspondingly, after optimizing for the gamma values, we see that one gamma has a much lower value than the rest, meaning that that specific location does not contain a Gaussian UB for the bond fields. And yes, so here is roughly how the bonds look. They are depicted here with some sticks of different colors for different covalent bond types and the field values at their centers. And this is finally the complete molecular graph, which has been extracted by this procedure. So yeah, the full pipeline is sort of, you generate these field values at specific locations that you predefine. And after the values have been generated, then you need to extract a molecular graph in this kind of complicated way, but not really that complicated. So there are two sort of deterministic extraction phases and then subsequent optimization phases that happen here to extract the molecular graph.
Could you explain how you like initialize the whole process, like with the atoms and the bonds in this field? Like, let's say you want, I don't know, a molecule with like 6 carbons and 12 hydrogens. Like, how do you actually initialize the bond in the atom field to get something that sort of looks?
Yeah, so are you now talking about the generation phase where I'm actually generating the fields, or are you talking about the graph extraction phase? First, okay. So the first starts as any other diffusion model. You just simply have random noise. So each location in the 3-dimensional space is considered to be isotropic normal Gaussian. So the initialization is simply throwing a sample from a random isotropic Gaussian, putting that into the neural network that does many time steps of denoising, and then you arrive at a clean field, or hopefully clean enough field. And based on that, you extract a molecular graph here. So you cannot also enforce the model to generate 6 atoms. It will generate some number of atoms.
So there's no way to like condition it to
yes,
sort of
there
be,
is. There is. Yeah. So you can use classifier-free guidance, which Dani has very briefly mentioned. So yeah, I mean, you can do conditional generation. It's a conditional generative modeling problem. You can also condition on the atom types with atom counts, which we do, but it's not necessary, I guess.
Is another way to condition it just like how you train the model? So if you, I don't know, choose a certain class of molecules to train on, then it's going to spit out things in that class, I would assume.
Yes, of course.
Okay.
Yeah, so it should model the distribution that you're training on. Yes. Okay. Sorry. Yeah, so right here, I'm at exactly, I guess, that point, which is classifier-free guidance, which is one method to actually define conditional generation for diffusion models in general, not necessarily for this, but in general, it has been defined. So since many evaluated metrics are highly correlated with atom counts, we also consider actually conditioning the generated fields on specific atom counts. So now we actually try to enforce to a degree, a continuous degree by some parameter beta, that the fields will generate specifically some number of atoms. And yes, as we have seen, the point cloud methods are restricted to have a pre-specified number of atoms n, which they can move in space and denoise to some atom types. And to kind of determine how much the atom counts conditioning is, or how important this conditioning is to various metrics, we also consider this classifier-free guidance on atom counts. And we define specifically some variable z, which are binned atom counts, where each bin contains roughly the same number of molecules in the dataset. And these bins somehow resemble the rough number of atoms that should be generated, and the beta controls the guidance amount that we require. So how much do we want the generated fields to adhere to the correct number of atoms that we condition on can be specified by the parameter beta. Okay, so briefly for the benchmarking data that have been used. First, the smaller dataset, QM9, which contains molecules of 9 heavy atoms, or at most 27 atoms, including hydrogen. And there are 5 atom types and about 130,000 molecules. And one example of a molecule is depicted here. Then Geom drugs, it's a considerably larger dataset. This dataset contains many conformations of various energies for each molecular graph. And therefore, this number of conformation is a lot larger than the number of 300,000 unique, about 300,000 unique molecular graphs. And yeah, up to 181 atoms, I think, if you include hydrogen. And probably the average is about 40 atoms. And about 27% of those molecules are actually also chiral, so containing some chiral center. Briefly, we go now through some experiments. First of all, the atom and molecule neutrality defined as the percentage of generated molecules that are neutral and the percentage of generated atoms that are neutral. So the molecule neutrality is defined slightly differently in that it considers a molecule to be neutral if only all of its atoms are neutral. And then validity checks only that the molecules can be parsed by RT-kit, which generally checks for valency and whether the molecule can be calculized. Okay, so on the lowest row, we have the data kind of showing that on the QM9, the easier data set, roughly 100% of the molecules are neutral and valid. So meaning that larger numbers are just better on this data set. And yeah, we obtain very good performance. So outperforming pretty much, I guess, all of the other methods. And then for the Geom drugs, here there are many, many caveats that need to be explained. I guess any one method in this table cannot be compared strictly or reliably to any other method, almost, on this specific data set, because there are very many settings that have been used. So some models generate explicitly the aromatic bond types. Some other models use only 5 lowest conformation energies. We use 30 lowest as some of the first works on this. And some even used the lowest, kind of creating hugely different numbers of training molecules, which of course makes the comparison not super reliable. Then we look at a few molecule quality properties. So for this, we define the total variation between the count distributions of various atom types and covalent bond types. So the total variation is just some distance metric between distributions, between probability distributions. And then we also have the MST defined as the maximum similarity to test. So we take the Tanimoto similarity between the Morgan fingerprints, between the generated molecules and the test data molecules. So there is a subset of the data that is considered as the test. And then based on the most similar fingerprint, which is between 0 and 1, we measure the average maximum similarity to test of the generated molecules.
Could you briefly elaborate what you mean with similarity here again, and how you would measure this or score this? What's the metric?
Yes, so I guess it's the Tanimoto similarity between Morgan fingerprints, where the Tanimoto, if I remember correctly, it's just the intersection over union of the exact same fingerprints that are some Morgan, some representation of molecules. So a very simplistic representation of molecules, and then we take all of the generated molecules and compare them across all of the test data molecules, and then kind of match to the best match. So in a perfect scenario, I guess you would assume that this number should go to 1, or very close to 1.
Does some of these metrics also cover how well you cover the landscape of all the molecules in your reference?
That is a good point, yes. So especially TVA and TVB, and then there are a couple of other metrics, I guess, in the dataset that consider mode coverage. But the idea is that they also consider, as Martin said, not only how high quality the molecules are, but whether you cover the whole distribution that you are training on. So that was a good point. Okay, so here we obtain roughly competitive performance, except with a big exception on the total variation of atoms. And then we reiterate how essentially point clouds are hard constrained to generate a specific number of atoms for each molecule. So because of this, you can of course trivially match the atom counts of the data, and then of course the various atom type atom counts will probably have like a much better TVA, but this is kind of just some property of point clouds. And then similarly on geom, but yes, again, with all the caveats that have been mentioned, with different data settings, different bond representations, and so on. So yeah, the conclusion would be that the method is having quite high performance, and then as we will see later, can also nicely capture chiral molecules or distinguish enantiomer pairs, which is quite nice. Okay, now before that, I will briefly go through confirmations. So before the chirality part, I will briefly go through the confirmation metrics. So the first one is quite easy. It's the distribution of the generated bond lengths. And this we measure in picometers because the models are generally so good in generating confirmations that, yeah, you actually need to measure them in picometers and not angstrom, which is the scale that you usually use for bond distances. And then, yeah, we just achieve surprisingly good performance. Even though we don't directly model the coordinates, we are still able to achieve really, really good Wasserstein distance, which is, well, another metric for distances between two probability distributions. And the same for bond angles now measured in degrees. So on QM9, about 1 Wasserstein distance measured in degrees, it's looking quite good. And similarly for geom, it doesn't become that much worse with the bond lengths and bond angles. So those were the simpler metrics. And then we also look at energies, conformational energies. And the first one is molecular force field energies, which directly or explicitly consider covalent bond types. And since many of the methods explicitly consider covalent bond types, we also include this metric for field for conformational energies. And then finally, XTB Wasserstein distance. So energy is computed with the same tool that has been used to optimize, or the same tool that has been used to optimize the conformations of the data that we are actually using for the training. So again, roughly very good conformations, given that there is no explicitly modeling the atom coordinates. Yep, so this was the point. Okay, so now the chirality experiment. We look at a very simple small 48 molecule data set where all of the molecules are chiral. So they have a chiral center. And we specifically have a carbon connected to another carbon, nitrogen, oxygen, and hydrogen. And then the way the chirality can be determined, as Dani presented earlier, is with some function f, which is the determinant of the kind of, well, it's the signed volume of a tetrahedron. So the chiral tetrahedron, if you look at the signed volume, you can actually strictly determine the chiral configuration. Well, that coupled with the atom types and the residue types, essentially, can tell you directly what the chirality is. But what this experiment shows is that on a skewed data, so the data is shown in blue, the data distribution is shown in blue, it's skewed towards one of the enantiomer configurations. And we try to find which methods can capture this. And first, in orange, we simply have an empirical confirmation that essentially a molecule and its reflected counterpart will have an equal probability. So you can say the equal probability on the two modes. And yeah, this essentially just confirms that the enantiomers are non-distinguishable for IFE invariant neural network parametrizations. Then we actually surprisingly look at GDM-AUG, which is a non-IFE invariant point cloud method, and it seems that point cloud methods still seem to suffer on a chiral data set. So even though now clearly the model distinguishes the two enantiomers, there's some difference in the generated distribution for one of the enantiomers. So the left one has fewer, which is correct, and the right one has more generated molecules. Still, there is a middle zero volume tetrahedrons that are generated, which are, of course, are unnatural conformations, and they seem to have this sort of problem when directly modeling specifically a chiral data set. And then we finally have fields which seem to be quite appropriate for this task, where FMG in red captures this distribution quite well. Okay, so I will now briefly go through conditional generation, where we consider two different properties. And the setting is that we have 50% of the QM9 data as the training data and 50% as a training data for a discriminative model for the same property that we train the generative model. And these are the two models for alpha polarizability and delta epsilon, the HOMO-LUMO gap for the orbital energies. And then we correspondingly have two classifiers determining what polarizability and HOMO-LUMO gap a molecule has. And then we measure how well the molecules adhere to the property Y that we use to condition using the phi Y. And Y is either alpha or delta epsilon. And what we find is generally a reasonable adherence to the conditioning property, but mentioning here that the model seems to trade kind of this rough measure of stability on the QM9 dataset, which is neutrality, for adherence to the conditioning property. But in general, quite reasonable. And then it's also quite nice here to mention that since we do not need to explicitly generate a specific number of atoms, this would probably be quite useful in general for conditional generation of molecules with some specific properties. So if we want to have many properties to condition on conditionally generate molecules on a vector of multiple properties, that vector of multiple properties in point clouds would have previously needed this joint distribution of the properties and the atom counts, which of course cannot necessarily be so easy to determine based on some data set. But here, yeah, you don't need this. And then we show this. So beta equals 0, FMG beta equals 0 is a model that is not conditioned on atom counts and can still conditionally generate some molecules.
Could you briefly say again which data has been used here, and in terms of absolute abundance or absolute number of inputs, roughly estimate how big was your training and test data set?
Yeah, so it's QM9, and I believe it's the training subset, which is 100,000 split into equal parts. So 50,000 for training the generative model and 50,000 for training the discriminative models. So the test here is the discriminative model telling whether the generated molecule is correctly generating according to the conditional distribution property. Yeah. Okay, now we briefly show some ablations. So some of the design choices that make our performance competitive. So we first look at removing the reference rotations, which have been presented as a way of mitigating the rotational invariance problems, and then, or the lack of rotational invariance problems. And then FMG AUG is a model that receives randomly rotated molecules as training input. So before we create the field, we randomly rotate the molecule. And then this, for instance, has a molecule neutrality at 82.6, whereas adding the reference rotation, so FMG beta equals 0, the number becomes 92.2. This is by far the most important design choice. And then we also have the atom count conditioning, which is quite important for TVA, but roughly doesn't change that much any other metrics. So other than atom count metrics, of course, conditioning on atom counts doesn't change the quality of the molecules as much. And then finally, this is sort of an empirical confirmation of how nice our conformations are. So even when detecting bonds based solely on the atom distances, we are able to achieve a neutrality of 91%. So the bonds here are detected based on some intervals, some reasonable intervals where a covalent type, bond type 1, for instance, between carbon and nitrogen should be. And yeah. So I was going to go a bit through scaling, but I don't think there is that much time. I guess the bottom line here is that it is probably about 3 to 4 times slower during training compared to one of the simpler point cloud methods, EDM. But then still manageable, especially in terms of memory. And then the generation speed is on par, if not even better, for very large molecules. So molecules of 180 atoms or so. And then, yeah, to summarize, yeah, fields provide a pretty promising molecular representation. And then we have seen how point clouds are unable to distinguish between an enantiomer if they are free invariant, and furthermore, adaptations that are chiral aware are arguably prohibitive. And then surprisingly, point clouds in general might have problems with chirality. And then that their flexibility allows conditional generation without restricting or requiring the atom counts. Reference rotations keep our performance highly competitive, so this is a very important design choice. And for future directions, the main point is the size of U, which we consider to further optimize through various, there are various options here. So the first idea we have is to perhaps have sparse or uneven set of points G, where we evaluate our fields. And of course, this would probably require a different parametrization. For instance, attention parametrization is quite well suited in this case. A more coarse resolution, the latest kind of results or experiments showcase that it's probably possible to create an even more coarse resolution and have the fields become smaller, thus allowing deeper neural network architectures and probably better performance in fitting our data sets. Or latent diffusion, so representing the fields as some latent representation that is of much lower dimensionality, and then creating the diffusion model in that space. And then conditioning on multiple variables is probably a very nice experiment and probably quite difficult to do in point clouds since they require the atom counts. And finally, pocket conditioning, which is probably possible as opposed to, for instance, protein conditioning. So full protein conditioning in fields is probably prohibitive. But pocket conditioning, given that if we want to generate binders for a specific pocket, that would be quite interesting, especially given the chiral sensitivity of the model.
My talk focuses on latent variable models. And the goal is to give a kind of an overview that how structured variants of those latent variable models can be used for quite many different prediction and optimization tasks. So Dani's and Alexandru's presentation focused on generation using diffusion models. I have a little bit more on sort of a modeling focus in my talk, not just generation. Before I start, I just very briefly listed what our research group really does. So overall, the work is divided into sort of a probabilistic machine learning, computational biology, and then applications. And the talk that I have prepared for today sort of covers many of these topics. and we are really focusing on nonlinear or deep latent variable models, which are generative models of this type. So we have a latent variable generated from some Bayes distribution, typically a Gaussian in standard models, and then the actual data item conditional or the latent from some other distribution, which is typically from the exponential family. The specific feature of these models is that the parameters of the exponential family are obtained by a nonlinear mapping D, which takes the latent as the input. So standard deep latent variable model. Once we observe data, Y, we become interested in the posterior of the unknowns, which are the latent variables. Or if we don't know the model yet, we would like to use maybe the evidence or the marginal likelihood for training the model. Unfortunately, both of these quantities are intractable. So in machine learning field, a common approach is to use the auto-encoding variational Bayes, which means using amortized variational inference. So we estimate the true posterior distribution of the unknowns with some parametric distribution Q. And amortization means that we have another mapping called encoder function that maps the data item into the parameters of the variational approximation. And if these mappings, phi and psi, are neural networks, then we are talking about VAEs. All right. And to make these models a little bit more useful in practice, we are primarily focusing on conditional generative models. So we always condition the generation with some covariates X, both the generation of the latent and then the generation of the actual data item. And then the amortized inference also sort of have access to these conditioning variables. Sort of the same setup, but now we add the conditioning terms as well. And there are different parametrizations for the conditional modeling. So we can define the model such that both the prior and the decoding likelihood see the covariates. or such that only the likelihood or only the priors is the covariance. And we are primarily focusing on the last parametrization. All right, so I'll try to cover 3 topics which sort of address the limitations or in some cases also possibilities of these VAEs and conditional VAEs. The first focus is on the fact that the data samples are completely independent in the standard models. And why correlations are important, and I thought about maybe highlighting the longitudinal or temporal data as an example case, because that's sort of the most obvious reason why to bother modeling correlations. So longitudinal designs are specific type of time series data sets that consist of multiple subjects, and each subject is then measured repeatedly over time, as sort of illustrated here. And this experimental design sort of implies that there are correlations both within a subject over time, as well as correlations across multiple subjects. And then if we want to use these kind of latent variable models for this type of data, we would like to be able to model these correlations, because otherwise, yeah, it's a kind of a nonsense. There are many ways to implement the correlations in these models. On the probabilistic modeling side, I think the Gaussian process approach is probably the most commonly used. And what it means is that we simply assign the L-dimensional latent variable Z to have a multi-output Gaussian process prior. Sort of a kind of a technically, we can say that Z is now defined as a function of X, the covariates inputs, and then we assign this F function a Gaussian process prior. And this Gaussian process prior typically has a 0 mean, and then it has, in this multi-output case, a cross-covariance function, which measures the smoothness of the correlations and dependencies in the latent variable. And then what this means in practice is that now if we take any finite collection of inputs, let's say n data points or n prediction variables, then the corresponding latents have a joint Gaussian distribution, correlated Gaussian distribution. So the latents across these n inputs then have a joint Gaussian, in this case a 0 mean, and then a full covariance matrix defined by the cross covariance of the Gaussian process prior. And the sort of the intuition is that the cross covariance function sort of defines that how strongly the different data points are coupled. So for those data points where the inputs are close by, then the correlation is high. For those inputs where for those data points where the inputs are far away, the correlation is low. And then of course that strength of the correlation is also learned from the data. So this has been proposed already a while ago. What we have been primarily using is this kind of additive variant of the model, so that the latent variable is defined to be a sort of a sum of GP priors, as denoted by here, such that each of these functions has a separate Gaussian process prior. And kind of the benefit of this kind of approach is that it sort of allows us to have some sort of interpretability of the latent variables, because we can now automatically factor them into additive terms, much like in linear mixed modeling approach. And then the conditional generative model becomes the following. So now the latent variable, it doesn't have the standard IID Gaussian, instead it has the Gaussian process prior, which implies correlations. But again, remember that this is in real life, this is a normal distribution, correlated. And then the distribution for the data items remains the same, decoder-based exponential family distribution.
Are the multiple kernel functions here operating on different dimensions of the covariates?
Yes. Yeah, thanks, Martin. So indeed, the way we typically implement these models is such that each function depends only on a subset of the covariates, and then that provides the interpretation. And then I use again the longitudinal data as an example. So because now much of the sort of the magic of these techniques builds on the choice of the covariance functions. And for example, in the case of modeling longitudinal data, we can use, for example, the exponentiated quadratic kernel to model shared effects shared across all the subjects. Category effects we can model maybe using a product kernel of the zero sum and then the EQ kernel. And then we can also have non-stationary effects, such as one may expect in the case of rapidly happening events like disease onset. And then we can have a ID-specific random variation using the product of the ID and then the EQ kernel again. And then we sum them up. So again, this is just the one dimension of the latent space. Of course, the latent space has many dimensions. For visualization purposes, I'm only showing one. And when we add the kernels or the corresponding latent variables, this is then the final latent variable that we have. But now we can automatically decompose it into these interpretable terms via the additive feature. Okay, and then a couple of more details before I show how these things work. So let's say that we have data set XY, size n. So we have n many data points, the covariates or the inputs, the corresponding data items, and then the latent variables, which we never observe. And then we are primarily first interested in the marginal likelihood, because we want to have some sort of object or objective to train the model. So we can choose the parameters and hyperparameters so that the marginal likelihood is maximized. And exactly the same way as for the standard models, we just integrate out the latent variables to get the marginal likelihood. And the only difference here is that now while the likelihood term factorizes, then the prior does not, because we have constructed it in a way that it captures the correlations that we want to model. Okay. And then again, these models are really not tractable. We don't have analytical solutions, so we need to rely on autoencoding variational Bayes. So we approximate the true posterior of the latent variables with the variational family. In most of the work, we are using mean field approximation, where we have an encoder that maps each data item into the corresponding parameters of the mean and variance of the latent variables. And then these functions mu and sigma are the typical neural network encoder models. And then we can use, for example, the Aalto objective to have a lower bound for the marginal likelihood. And then again, it has the same form as the Aalto typically has. I have written it here in a typical two-part way, so that the first part shows the reconstruction loss, and then the second part shows the regularization KL divergence term. The first part is easy to compute because these elements factorize according to the data points. The second part does not, because we have constructed it in a way that it contains the correlations that we want to model. This has a closed form solution because everything is Gaussian. If you just have million data points, then it's a million times million matrix. So this can be slow to compute. So we have some theoretical results from earlier work where we proposed a provably tighter evidence lower bound for the evidence for a longitudinal or additive CP models, and then that provides us an upper bound for the KL term here, so that we have an objective against which we can train these models. And then we also have a kind of a novel minipatch compatible KL upper bound. Okay, I'll show first one simulated dataset, and then the point is to sort of demonstrate that what these models can do. And we took the MNIST images, 1,000 different digits. Those now represent our patients. And then 20 different time points are obtained by rotating and shifting the image according to the covariates associated to each subject or patient. And then we have an additional 100 test subjects, such that we are given the initial data points. And then the goal is to predict the remaining time points that we have not seen for the test subject. And this is an illustration what, for example, our model can do. And we have compared our model against many competing techniques, some of which use also Gaussian process-based techniques, some use RNNs, some use GANs. And in many cases, it seems that our additive CB-based VAE model is a good choice and highly performed. I'll show you a couple of examples for real data sets a bit later, for real electronic health record data sets. But then I thought that maybe I saw this sort of a preliminary results from a time series single-cell RNA sequencing data set that we have been working on. So as you all know, time series.
Can I ask a question just
Yes.
on the previous slide? So when you did the simulation, like, how did you decide what correlation structure to use across, I guess, the time points and these patients who are just spitting out, I guess, numbers? And then does the complexity or simplicity of the correlation structure change how much data you need to train a model?
So the first part of the question was that how we choose the correlation structure. So I think in this case, we can't really interpret the model as a just black box, but we need to make some modeling choices. And then I think a natural way to do these modeling choices is based on the covariates that one has available. So we are modeling the effects of the covariates in the latent space. And in this case, I don't remember exactly, but I suppose we had 6 or maybe a bit more effects in the latent space.
Okay, so I guess that answers my other question, which is just like you have more covariates and you just need more data, I guess.
Yes.
Yeah.
Maybe also related to that, but in the modeling, how is that affected by multicollinearity? So for example, if you have age and blood pressure or something like this, you would already assume that those correlate with each other. So how would you deal with that?
Yeah, well, I don't really have a good answer, but I agree that it would complicate things. And then obviously one can do some sort of a covariate selection that which are really important, but we haven't really gone to that direction too much. That's a good question though. So another application is modeling time series single-cell RNA sequencing data. And so what this kind of experiment gives is a large number of high-dimensional observations, YTC, of individual cells, C at different time points T. And then the kind of the key feature of this modeling technology is that you can measure one cell only once. And then what we see is only these sort of a marginal distributions at different time points. But nonetheless, we thought that we want to try these our models on this type of data as well. We can't really apply the models directly as I explained a couple of slides ago, but what is typically done in the field is to use some sort of a earth moving distance or Wasserstein distance to match the predictions, model-based predictions with the data. So we have the data points, n many cells at certain time points, corresponding predictions, and then we can use the squared Euclidean loss to get the Wasserstein 2 type of distance measure between the predictions and the data points. So what we can then do is to train the kind of model that I explained earlier, but replace the reconstruction loss in the evidence lower bound with the corresponding Wasserstein, such that we still get the predictions from the kind of estimated latent variables, but we match the predictions with the data only via the Wasserstein matching between the individual cells. And this is something which is not finished, but I thought that it's kind of interesting. I wanted to present that anyway, and we have tested this on a couple of different data sets. So here are some data sets used in other computational works earlier from zebrafish, Drosophila, and then mouse. This many cells, this many time points, and then we can try to evaluate that how well these kind of latent variable models are able to interpolate between the time points. So we kind of intentionally leave one of the time points out and then see how well we can reconstruct the missing time points using our models. And seems to perform relatively well, at least against the previously proposed models, which are mostly using neural ODE type of techniques for the modeling. And here are the corresponding predictions. So this is the data color-coded according to time points, and then these are the predictions from our model in, again, 2D embedding space. Okay, before going into this electronic health record type of analysis, I just want to mention that many real world datasets contain missing values. So any subset of the features or the covariates may be missing completely at random. It's sort of trivial to handle the completely random missingness from the features, but less so from the covariates. And then the goal is to learn conditional VAEs from partially observed datasets that contain missingness also in these predictor variables X. And we do that by augmenting the generative model with the prior for the covariates as well, such that the joint distribution for the data, latents, and covariates factorizes accordingly. And then I just note that in this case, the covariates contain both discrete and continuous variables. So it's not quite exactly the same as typical VAE setting where everything is continuous in the latency. And I will not go into details, but the concept is sort of quite simple. So we extend the amortized variational approximation also for the unobserved covariates X. And then we maximize the evidence lower bound such that we simultaneously marginalize out any uncertainty in these unobserved covariates. And in terms of ELBO, it leads into this kind of objective where we have the typical reconstruction loss and then the KL divergence. The KL divergence just happens to have now is a two-part expression, one for the missing covariates and then one for the latent variables, which involves an additional expectation. And here is one example how this kind of a model works. So we took some observational longitudinal data focusing on Parkinson's disease, follow up about 5 years, 500 patients, about 3,000 samples, relatively low dimensional in this case, 42 features observed, 7 covariates available, but now it's a real world data set, so missingness in the covariates is something between 2 and 32, so quite high missingness. And then we apply these techniques that try to handle the missingness in the prediction variables as well, and then evaluate using a future prediction task. So we are given a data set and then additional test subjects such that we have the initial trajectories of the test subjects, and then we would like to predict the future Y values given the partially observed covariates X0 star. And I don't have a comparison across many different techniques. I'm just showing that how this kind of a technique that handles the missingness in the covariates performs. So the red bar is something where we just don't care. We press enter and hope for the best. The orange bar shows the performance using the commonly used mean imputation for the covariates. This is a KNN imputation, and then this is our approach that handles the missing covariates variationally as well. Okay, and then, so now our models can handle completely at random missingness in the features and the covariates. So we have the partially observed covariates, correlated latent variables, and then partially observed observations. We can easily extend them to handle structured missingness, which is missing at random or missing not at random. Quite a realistic assumption in medical datasets, for example. And that happens by adding an additional latent variable that models the missingness pattern as such, and then connect that to the missingness mask, which then implies that which data values are really missing. We can also extend the models such that they model the marked point process. So also the measurement time points are random variables. And we can do that using this non-homogeneous point processes or self-exciting Hawks processes or whatever we want to use. And then that happens before the actual modeling of the high dimensional data. The play diagram looks a little bit more complicated, but still sort of a straightforward. I will not go into details of how these are implemented. I just want to illustrate what these models can do. So this is again a simulated data set for simplicity. This is a kind of a real data set, again the MNIST type of longitudinal data, and then this black box now denotes the missingness mask, which we can model separately. And some of the missingness patterns also depends on the strength of the underlying signal. So the stronger the signal in the digit 3, the higher probability that the pixel values are also missing. I skip the empirical evaluation, but it performs really well. Okay, so that's the part 1. So I try to illustrate how these structured, meaning correlated latent variables, can be useful for longitudinal modeling, and illustrated that using electronic health records type of data, and then also something for the time series single cell RNA sequencing. We have also used these for survival modeling in a longitudinal setting. We have also changed the CP prior with a linear mixed model prior to get a sort of a similar performance. And then we have some work which sort of tries to improve the underlying methodology so that the performance improves across all these methods. But it's still sort of non-published.
Maybe one question about the intuition here. So it used to be so that in the old days, we used these simple imputation methods just to be able to use more data in our models. But it seems here that also modeling these intermediate relations between the covariates has some sort of benefit on your downstream tasks of interest. Is that the correct way to think about?
Exactly correct. Yeah, so I would claim that in most real data sets, there is some correlation between the missingness and the statistical properties of the model. But that's the way it's be done. Okay, the second part will be a little bit shorter, but focuses on using these latent variable models for Bayesian optimization. And you may be familiar with the concept of latent space Bayesian optimization. So it's a kind of a way to do black box optimization of high dimensional or structured objects. Why? By combining latent variable modeling, typically variational autoencoder, as well as Bayesian optimization for some specific feature C using GP surrogate in the latent space. And this is a figure from one of these early studies where they used exactly this technique for optimizing molecules. So you can sort of think about the molecule being encoded into latent space representation. And then you do that for many molecules and implement then the CP-based surrogate Bayesian optimization in the latent space. Use some acquisition function to choose which molecule to quantify next, and then do the experiment and feed the data back in and continue. And then obviously the actual molecule that you want to generate next is then obtained via the decoder from the latent space. And our hypothesis is that maybe these conditional latent variable models, Gaussian process prior VAEs in particular, can provide a better structured latent space for Bayesian optimization. So our approach is something where we do latent variable modeling using a conditional CP prior VAE, where we have, let's say, the molecules Y generated from the latent variable Z. But now the latent variable would not be independent, but depend on exactly the feature that we would like to optimize, as well as possible additional features that we know of these molecules, as an example. And then apply the standard Bayesian optimization in the latent space that we obtain from this kind of a model. And one needs to keep in mind that in this kind of a setting, the covariates are then only partially observed. In a typical Bayesian optimization setting, you have only a handful of data items for which you know the quantity that you would like to maximize or minimize. So this is only available for typically a small number of data points that you want to do the modeling. And another special feature is that we may have additional properties known of these data items called X in real world applications. This is typically a kind of an approach which is not really commonly used in Bayesian optimization, but I think one should, because there's possibly information in these X values that tell something about the C as well. So this is sort of the approach that we have proposed. So now illustrated in the context of molecule optimization. So the sort of the middle part is similar with the existing techniques, except that when we represent the molecules in the latent space, we do that by assigning a latent variable, this correlated prior. This correlated prior is the same additive CP that I used earlier, such that now it depends on the features that we may have partially available from different molecules. And the hope is that now the latent space is automatically shaped according to these predictor variables C and X. And then hopefully it makes it easier to predict the X or optimize the C once the latent landscape is shaped in a hopefully optimal way. Okay, so the pseudo algorithm is something that we train a CP prior VAE with partially observed covariates. C and X may or may not be partially observed. Y values are fully observed. We obtain the latent embeddings Z for these data items. We carry out the Bayesian optimization in the latent space. We have been using the expected improvement acquisition function. One can use also other acquisition functions. Then we decode the chosen data point Z prime to an actual data item Y prime, evaluate the black box function to get the C prime, and maybe also X prime if that is available. Feed that again back to the Bayesian optimization loop and continue. And here are a couple of examples how this works. So the first focus is on generating molecules using the junction tree VAE. This is probably not the state-of-the-art technique for molecules anymore, but it doesn't really matter. We are giving the all models the same VAE architecture and then only changing the way we treat the latent variables. The data is from this zinc 250K, and we assume that only 1% of those have been observed. So we have the goal is to optimize the so-called penalized log P quantity of these molecules, and then only 1% of the molecules has this feature known. We are using this penalized log P as 1 hour of covariates, as well as additional 5 features which we can easily compute from the molecules that we are modeling. And this is sort of the part which is now hopefully giving us a benefit in this Bayesian optimization. And here is a comparison of different techniques over the number of Bayesian optimization steps that we take, and then the penalized log P score on the y-axis, and the purple line is the performance of our model. So maybe not outperforming, but I would say that the competitive performance relative to the other techniques. Another example using generating single variable mathematical expressions using grammar VAE, so something kind of maybe complicated if you think about optimization in continuous domain, but again, the VAE architecture gives us the latent variables, and then the Bayesian optimization is implemented in the latent space. So the target is to find this kind of expression, and the objective is the MSC of this function in a certain range of inputs X. And sort of the key innovation of our technique is to use this C value as one of our partially observed covariates, as well as other covariates that we can easily sort of obtain from these expressions that we are modeling. And again, many different techniques compared. The purple one being here our model that again performs quite competitive. All right, so that was the part 2. That's the only sort of finalized work that we have been able to put together for the high dimensional Bayesian optimization, but there's a sort of more in sort of planning at the moment. This will be also presented after a couple of weeks together with Alexandru's and Dani's work in the same conference. And then the final part, I think I still have time for that, focuses on the fact that these latent variable models cannot really model temporal or dynamical systems. Although it turns out that they are, of course, easy to extend the direction as well. And the problem setting is something that we have high dimensional observations, Y, of dynamical systems, of a dynamical system at arbitrary time points and across many trajectories. So time points, arbitrary, and then the corresponding measurements. And the measurements in this case could be, for example, images or single cell data as an example. And the goal is to model the data using a continuous time dynamics in a low dimensional space. Now the latent space here is denoted by X. And the latent space dynamics are now illustrated by this line in two-dimensional space, and these are the high-dimensional observations. This is something that we would like to achieve. Of course, there are many different techniques available already which can do that. We are using the latent neural ODE as our sort of starting point, where the initial state in the latent space is sampled from some prior distribution. We sample the parameters of the dynamics model, assuming that they are also modeled probabilistically. And then we solve the intermediate or later time points by solving the initial value problem of the dynamical system, essentially numerically solving the ODE system in the latent space, starting from the initial state all the way to the later time points, TI. For each of the measurement time points, we then decode the data using the decoder function with samples from the exponential family distribution. So the concept is shown here on the right. Initial state sampled from some phase distribution. Then we solve the initial value problem, or numerically integrate the ODE system, decode the latent states into the data. Okay, so that was actually proposed already at the time of the neural ODE paper, 2018, and then follow-up papers focusing on these latent neural ODEs. What is challenging with these techniques is that the training on long trajectories can be really slow and unfortunately unstable. So there's theoretical work for the discrete time systems saying that the sort of the objective function in terms of the Lipschitz constant can kind of grow exponentially in terms of the length of the trajectory. And that's sort of demonstrated here in this empirical evaluation. So the plot at the top shows the train loss over the iterations of the training procedure, where we double the length of the trajectory every 3,000 iterations. So we optimize first, double the length of the trajectory and optimize, and then suddenly after a certain time point, the optimization doesn't really work anymore at all, demonstrating that the optimization simply becomes unstable, most likely due to unstable gradients of this model. The bottom figures illustrate what the loss landscape looks like at these optimized positions. And this is obviously just a one-dimensional projection of really high-dimensional optimization landscape, but sort of nicely agreeing with the intuition that with longer trajectories, the optimization landscape becomes highly multimodal. So we have been proposing these kind of multiple shooting-based approaches where we split the latent trajectories into short segments, and that's not a novel idea either. We would like to do that probabilistically though, so that we can still sort of train these probabilistic generative models. And for that purpose, we have been proposing these auxiliary shooting variables, S variables, which are these shooting points here in the latent space, and then a reasonable prior for these shooting variables, which has this kind of first order Markovian structure, such that the next shooting point is always close to the end point of the ODE solution that we obtained from the previous time. And then the generative model becomes something as shown here. So again, we sample the dynamics parameters, the decoder parameters from the corresponding distributions. We sample the shooting states from this continuity prior from the previous slide, and then always solve the intermediate points using the initial value solution of the corresponding ODE system, such that we start from the shooting variable and then integrate forward in the latent. And then decode the data items from the corresponding latent variables. That's sort of demonstrated here on the right. Again, difficult to handle anything analytically, so we are relying on amortized variational inference. So the target is this joint distribution of the dynamics and decoder parameters as well as the shooting states, given the observations of the trajectory. And then that is now approximated by a variational distribution, which factorizes into components corresponding to the model parameters and then amortized part for the shooting states. And then if you think about it sort of in terms of a VAE language, here is our generative model, and then now the variational inference of the shooting variables then gives you the kind of encoder structure for your model. Okay. So then the second important aspect is the specific type of attention model, attention-based encoder model to be able to implement these both efficiently and accurately. And we have been proposing the encoder which is flexible and efficient, such that it handles irregular time grids, so the time points can be arbitrary in your measurements, such that the encoder generalizes to new time grids at test time. It can handle long trajectories. It allows us to do parallelization, as I'll show a bit later, and then handle noisy and partially observed data. And the encoder has this kind of a three-step structure, where the first part compresses the high-dimensional data into some lower-dimensional representation. The aggregation part is implemented by a stack of attention layers using a specific type of attention technique, and then the last part simply sort of reads the parameters of the variation approximation. And if I have time, I'll just try to quickly illustrate that how this specific type of attention encoder works. So what is shown on the left is the standard dot product attention technique, where we take the dot product of the queries and keys, and then the corresponding softmax to get the weights for the values. We have been proposing a time-aware attention, such that we are only looking at a kind of a reasonable window around the current time point that we are modeling, using a time-aware attention mechanism defined by this formula, which is illustrated here. So that allows us to such that the attention model doesn't grow arbitrarily large while the length of the trajectory increases. We have also replaced the standard positional encodings with continuous relative positional encodings, so that the encoding of the position is now positioned at the time point that we are interested in modeling. And then again, evidence lower bound based training of the model. You don't need to care about the complicated expression. The point that I wanted to make is that, again, it has the same structure. The first two parts correspond to the reconstruction of the data from the latents, and then the four remaining parts implement the regularization. The parts which are highlighted in red involve the dynamics in the latent space. And And together with the multiple suiting and the factorized approximation for the suiting variables, we can solve all these two terms in parallel, which sort of increases the efficiency quite a lot. If you have a long trajectory, you just chunk it into small parts, and then the optimization of the entire trajectory can be run in parallel. And a couple of simple toy cases first to illustrate how this works. So pendulum is the first example, bouncing balls in a box. So the dynamics are defined by real physics, and then the observations are these high-dimensional image-based observations, and then the rotating MNIST as a second additional benchmark. And then this is just a quick note that how well our model can make the future predictions compared to some existing neural OD processes or OD to VAE models. And the blue one seems to perform quite well across these synthetic benchmarks. And this is sort of effectively what the model tries to do. So this is the data, high-dimensional trajectories. These are the corresponding predictions. And then we can also use these to somehow quantify the amount of uncertainty in the latent variables. So this is now the mock-up data from the Carnegie Mellon University, where a person is wearing about 100 sensors at the joints, and then it measures the 3D position of the sensors, and then the goal is then to model the dynamics. And obviously that's difficult to do using parametric models, but it can be easily done in latent space dynamics models. In this case, the visualization is done by using a 3D latent space, where we implement the kind of a latent neural OD system that I proposed. And then starting from the initial state, we sample the parameters and initial states multiple times, and then you can interpret these lines as samples from the posterior, sort of demonstrating how the amount of uncertainty increases while the person walks further to the future. And this is the corresponding then reconstruction in the actual data space. Okay, and then now moving back to molecular biology examples. So the same kind of a setting that I already explained. So modeling now transcriptional dynamics using single-cell RNA sequencing. The setup is exactly the same that you are very well familiar with. So high-dimensional observations, YTC, of the individual cells at individual time points, and each cell is measured only once. So we can't really use the models directly, as I explained, but again, the kind of earth-moving distance-based measures to kind of connect the predictions with the data are really useful. And motivated by actually many of the existing techniques, we just wanted to test how well our models would perform in these kind of tests by training our latent neural OD using the single cell data, and now changing the loss to be completely based on the Wasserstein based loss in the data space as well as in the latent space. So this is also something that is not finished yet, but I just show you the kind of preliminary results. And this is visually what is happening. This is not exactly correct, but the predictions are now the individual cells at different time points, which we connect to the actual observations using the Wasserstein 2 measure. We implement the same Wasserstein loss also in the latent space, but I was not able to put that in this figure, so sorry for about that. And then we took a couple of data sets from mouse and zebrafish, and then simply compared our modeling tool with one of the, I think, state-of-the-art models called SC Node. And then these are the Wasserstein distances for later time points in this time series, given the initial part of the data, and then how well we can predict the population at later times, and measure the performance via the Wasserstein distance.
Harri, what trade-off do these multiple shooting points bring? Because let's say we want to model the dynamics all the way to x5, starting from x1. But do we need this as intermediate measurements that we need to take?
Yeah, that's a really good question, and then I think that in this particular example case, maybe the multiple shooting is not the key determinant of the performance. But we have been also trying a kind of a different variants where we model both the sort of indirectly the splicing as well, which seems to further improve the performance a bit using the intronic reads as well. Yes, depending on the length of the trajectory, the importance of the multiple shooting becomes more or less significant. And here is the kind of the illustration for what the true data looks like when the data points are color coded by the time points, and then the predictions, and then for the mouse data, it seems to be performing visually better, whereas for the zebrafish less so. But again, this is not yet finalized, so something that we are still working. I want to conclude with a really quick note that we have been also extending these techniques to spatiotemporal processes. So the dynamical systems do not need to depend only on time, but maybe also on space. And then we are talking about partial differential equations. And we have been extending these latent variable models to sort of learn space-time continuous neural PDEs, again from partially observed states. And here are some sort of illustrations of the physics systems that we have been considering. We have also tested these techniques on some spatial transcriptomics datasets, but maybe I can't really report anything on those yet. And then kind of the last model variant is that we have been also extending these to kind of really extreme cases where we are interested in modeling spatiotemporal dynamics as well as point process observations. So in a typical setting, when one talks about the spatiotemporal modeling, the measurements come from some sort of a fixed grid. But we have been considering a case where the measurement time points as well as the locations are also random variables, and they depend on the underlying dynamics. And this is something that Valerie will also present after 2 weeks in the same conference. So that was the part 3. So we have been working quite a long time on these latent variable neural or Gaussian process differential equation models. And then lately being also focusing on modeling transcriptional dynamics using these kind of techniques, both using dynamics model as well as the correlation models that I explained earlier, and we would be happy to discuss more about those if you are interested in. And then extended these techniques to partial differential equation type of systems. Would be really cool to try these on spatial transcriptomics if you have temporal data from those kind of systems. And then ultimately we would be interested in controlling these systems. But this is something that we haven't really had time or success so far. But this is the only application that we have been able to put together so far.